Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Amaida Amir and I am Assistant Professor in Department of Anatomy. Today our topic of discussion is the formation of the heart tube. Before the formation of the heart tube, you must know the structure of the embryonic disc. In this structure, there are two openings in this embryonic disc. One is cranially and the other is caudally. This is the cranial and caudal end of the embryonic disc. Now, this opening is called oropharyngeal membrane and this opening is called cloacal membrane. In its lower central part, there is formation of a structure which is called primitive streak. At the upper end of primitive streak, notochord will form. This whole structure is called neural plate. Now from this primitive streak, the different type of mesodermal layers will take origin, and they are the lateral plate mesoderm, intermediate mesoderm and the far axial mesoderm. The mesodermal cells from both the direction, they will start moving above the oropharyngeal membrane, and here they will segregate to form cardiac mesoderm. Initially the cardiac area is above the oropharyngeal membrane. As you know that mesoderm is divided into two layers. One is called the somatic layer and other is called splanchnic layers. Somatic layers form the body wall, the splanchnic layers form all the relative structures. The same will happen at this cardiac mesoderm. Here again the mesoderm will divide into two layers, somatic and splanchnic, and all the structures of the heart will form into the splanchnic mesoderm. Around this mesoderm there is formation of a structure which is called the heart field. So we divide the heart field into two parts, primary heart field and secondary heart field. Primary heart field forms of both the atria, the left ventricle and some part of the right ventricle, while the secondary heart field forms the right ventricle, the remaining part from here, the conus cordus, which is divided into the bulbus cordus and truncus arteriosus. The truncus arteriosus form the outflow tract of both the ventricles, right and left, and the bulbus cordus form the atria and the pulmonary trunk. Now the question is how the heart fields are formed. It's a term which is called cardiac precursor cells. These cardiac precursor cells have different sites of origin. They take origin from the primitive streak. From the primitive streak, these cardiac precursor cells will move up to the cardiac mesoderm from here and then from here. And when these precursor cells move here, they will start incorporating or invaginating or proliferating over here, and all these cells form the heart tube. So the two heart fields are formed, which I told before, the primary heart field and the secondary heart field. These heart fields will form in this cardiogenic area. Consider this is primary heart field and this is secondary heart field. Primary heart field and secondary heart field. The cell for the formation of primary heart field will move from primitive streak, and a primary heart field will form like this. And the cell for the formation of the secondary heart field will move from splanchnic mesoderm, and the cells move from splanchnic mesoderm. And beneath the primary heart field, a secondary heart field will form. This is primary heart field and this is secondary heart field, and all this is happening over this cardiogenic area. So after the formation of the primary and the secondary heart fields, there is the formation of endocardial heart tubes again in this area. Now the question is how the endocardial heart tubes are formed. Consider this box as the cardiogenic field or the cardiogenic area. In this cardiogenic area, there is formation of the cardiac primordium, and at the same time all the other structures are formed over here, which are the primary and the secondary heart fields. This black dot is called the cardiac primordium. Initially, this cardiac primordium is single, but with passage of time, it will appear in the form of the pair, and these pair will give rise the endothelial heart tubes, which are also called the cardiac tubes or heart tube. So the cells will proliferate here and form the tube-like structure. Now the embryo starts folding. The first fold is at the head region. Consider the top of this page as the head region. The second fold is at lateral regions, this region and at this region. Head folds itself like this, As you know, initially the heart lies at the cervical region, but with the head folds, the heart will come downwards into the thoracic region. Now the lateral folding of the heart tube takes place like this, and the two heart tubes, one and the other, that are separated from each other, they will come close to each other. But again, there is a complete septum between both of them because of their separate walls. With passage of time, this septum will finish, or this septum will disappear, and the heart tube will form like a single structure. Now the heart tube will be like this, and this is the upper end, cranial, this is the lower end, caudal, or this is cephalic end, and this is caudal end. Now there is formation of the pericardial cavity after the formation of the heart tube. The pericardial cavity will form in this cardiogenic area as an intraembryonic coelom. Coelom means a cavity, and this cavity is separate from the heart tubes. The heart tubes, initially they are two, when they will combine, they will form a single heart tube. Initially that heart tube lies outside the pericardial cavity. So consider this heart tube which lies outside the pericardial cavity, and this is pericardial cavity. But to move inside the pericardial cavity, there is the rapid proliferation of the mesenchymal cells, and these mesenchymal cells will bring this heart tube into the pericardial cavity, and that heart tube will move inside it. But some portion still lies outside and some portion lies inside. Consider this marker as a heart tube and this paper as a pericardial cavity. Dorsal mesocardium lies between the posterior part of this heart tube and between the pericardial cavity. With the passage of time, this dorsal mesocardium will disappear, and that will then form the transverse pericardial sinuses that we will discuss in our other lectures. The first layer which forms in the heart tube is endothelium, which is here called the endocardium. This flow chart will show you the formation of the cardiac muscles. The splanchnic layer, when it will start investing into the heart tube, which is already formed by the endothelium. This splanchnic layer forms the cardiac mesoderm. That cardiac mesoderm will form the cardiac muscles, which is also called the myocardial mantle. Now the two layers are formed, the endocardium and the myocardium. This is endocardium, this is myocardium. Between both of them is a very thin connective tissue layer which is called the cardiac jelly. This cardiac jelly, when will disappear, that will form the subendothelial connective tissue. Now the last layer, which is called the epicardium. The mesenchymal cells, which is actually the embryonic connective tissue, they will start moving towards the heart tube and form a layer which is called epicardium. At this stage, now the complete heart tube is formed with all its parts. One thing which is very important over here is that when the heart starts beating on day 23. Initially the contractions are irregular and they are not coordinated, but with passage of time they will become coordinated. So this is the end of our first lecture of CVS Embryology, and next time I will show you all the models and I will demonstrate them, the CVS on models. Thank you.